Hello and welcome everyone. Myself Mizanur Rahman, and today my topic of discussion is hemolysis and fragility of red blood cell. So today we will try to discuss about the definition, then process of hemolysis, then the fragility test, then condition when the hemolysis occurs, then hemolysis like chemical substances and substances of bacterial origin or substances found in the body. So this is our today's topic. And in the previous video, we have already covered the anemia. So if you have any doubt related to these topics, you can ask in the comment section. Now start. First of all, definition. What is hemolysis? Hemolysis is the destruction of formed elements. Now, what are the formed elements? The RBC, WBC, platelet, or everything are included in the formed elements. Now, in case of hemolysis, the main destruction is of RBC. And whenever RBC is destructed, there are release of one substance, that is hemoglobin. So hemolysis is the destruction of formed elements. To define more specifically, it is the process which involves the breakdown of the red blood cells or RBC and liberation of hemoglobin. Now second point, that is the fragility. Fragility is the process which is the susceptibility to be affected of RBC or to hemolysis or tendency to break easily is called fragility. Fragility means actually the broken tendency. And fragility is of two types. One is osmotic fragility, one is mechanical fragility. The osmotic fragility, which occurs due to exposure to hypotonic saline, and mechanical fragility, that is which occurs due to mechanical trauma, wound or injury. So one is osmotic, one is mechanical. So what is osmotic? Osmotic fragility occurs due to exposure to hypotonic saline, and mechanical fragility occurs which occurs due to mechanical trauma or wound or injury. Now, under normal condition, the only old RBC are destroyed in the reticuloendothelial system. This is the normal procedure. But the abnormal hemolysis is the process by which even younger RBC are destroyed in large number by the presence of hemolytic agents or hemolysis. So what happens? The hemolysis which is present, which will affect the younger RBC, and destruction will happen. Now this is the abnormal condition and this is the normal condition. Now the process of hemolysis. So what is the process or mechanism? Normally the plasma and the RBC are in the osmotic equilibrium. When the osmotic equilibrium is disturbed, the cells are affected. For example, when the RBC are immersed in the hypotonic saline, the cell swell and rupture by bursting because of endosmosis, and hemoglobin is released from the ruptured RBC. So what happens in normal condition? The RBC are present in the osmotic equilibrium in normally, and when the osmotic equilibrium is disturbed, some problems will happen. The cells will be affected. So if you take an example, when the RBC is put into the hypotonic saline, then what will happen? The RBC will swell up, and it will rupture. And whenever it is ruptured by bursting due to the process of endosmosis. Now what is endosmosis? We will discuss in the later chapters. And the hemoglobin is released from the ruptured RBC. So this is the process of hemolysis. Now go to the next point. Fragility test. What is fragility test? First of all, it is a test which measures the resistance of the erythrocyte in the hypotonic saline solution. So the saline will be hypotonic. Mainly it is taken as sodium chloride. Okay. And it may be taken at different concentration level. It ranges from 1.2 to 0.2%. And the solution at different concentration are taken in series of cones tube. And the one drop of the blood to be tested in the added to each tube. The sodium chloride solution and the blood in each tube are mixed well and left undisturbed for some time. And result can be analyzed by observing the tubes directly by centrifuging the tubes after 15 minutes. So what you have to do? You have to take the cones tube and take one drop of blood, then add some the hypotonic solution like sodium chloride solution, and then put it for some time to be rest, like nearly around 15 minutes. Then centrifuge this, and when you centrifuge, you can directly observe the result. So what will be the result? Let us see. First of all, direct observation. Number 1. If there is no hemolysis, fluid in the tube appears turbid. If hemolysis started, turbidity is reduced. If hemolysis is completed, fluid becomes clear. So what are the direct observation? If no hemolysis occurs, then the fluid in the tube appears turbid. It will be as it is. If hemolysis is started, turbidity is reduced, and hemolysis is completed, then fluid becomes clear. Let's suppose this is the 3 jars. In the first tube, in the second, and in the third. I have mistake done. Just draw it again. 1, 2, 3. This is the first one. This is the second one. And this is the third one. I have no white color, that's why I have to show like this. Okay, let us start explaining. So in the first one, what is happening? Here the turbidity is more. And in case of this point, there is no hemolysis. In case of second point, hemolysis started. And in the third point, hemolysis is completed. So these are the 3 conditions which you can observe directly. Now, observation after centrifugation. If in case of first point, if there is no hemolysis, cell sediment at the bottom with clear colorless fluid above. Second point, if hemolysis started, the cell sedimentation is less and the fluid becomes slightly reddish because of the release of small amount of hemoglobin from few hemolyzed RBC. And third point, if hemolysis is completed, fluid becomes more reddish without any sedimentation due to release of more and more of hemoglobin. More amount of hemoglobin from all the hemolyzed cells. So let us draw it again. So what happens if there is no hemolysis? 1, 2, 3 again. The cell sedimented at the bottom, and with colorless fluid above. There is no color. In case of second point, the cell sedimentation is less and the fluid color is slightly reddish. Why the color is reddish? Because of release of small amount of hemoglobin from few hemolyzed RBC. Now, in the third point, if hemolysis is completed, the solution is more reddish and there is no sedimentation at all because release of more hemoglobin from more hemolyzed RBC. So these are the points you must know. Now come to the index for fragility. What are the index by which you can know? After 20 minutes, no hemolysis up to 0.6%, onset of hemolysis around 0.45%, completion of hemolysis around 0.35%. And at 0.45% only the older cells are destroyed because their membrane is fragile. So these cells cannot withstand with hypotonicity, but younger cells are not affected. At 0.35% even the younger cells are destroyed. So what are the indexes? After 20 minutes, you can see that if no hemolysis up to 0.6%, the onset of hemolysis around 0.45% and completion of hemolysis 0.35%. Why completion of hemolysis at 0.35%? Because at 0.45% only the older cells are destroyed because their membrane is fragile. So these cells cannot withstand with the hypotonicity, but younger cells are not affected, but 0.35% the younger cells are also destroyed. Now what are the conditions? Conditions when hemolysis occurs. So let us start. There are 4 conditions which are mentioned in this book. So let us start. Number 1, hemolytic jaundice. Number 2, antigen antibody reaction. Number 3, poisoning by chemical or toxin. Number 4, while using artificial kidney or hemodialysis or heart lung machine during cardiac surgery. Part 7, when jaundice occurs in the hemolysis, and antigen antibody reaction and poisoning by chemical or toxin, and while using the artificial kidney or hemodialysis. So let us explain these conditions. Whenever a patient is affected by jaundice, and especially it is the case of hemolytic jaundice, then what happens? The RBC breakdown starts and the hemoglobin releases from that RBC, so there's problem can occur the hemolysis. And antigen antibody reaction. Suppose a patient who have done a accident, road accident, and the patient has lost some blood due to acute hemorrhage, and he has got the acute hemorrhagic anemia, which is a normocytic normochromic type, and then suddenly suppose he has the blood group A positive, and there is no A positive blood in the blood bank. Then what will you do as a doctor? You must find some donor who have A positive, but in case you do not find any A positive blood, or by mistakenly you have given B positive blood group into the A positive patient, what will happen? The antibody reaction will start and the blood will coagulate. Then the anemia also, hemolysis will start, and the breakdown of the RBC, hemoglobin will release from the RBC. So this type of problem may occur by poisoning or chemicals or toxins. If you eat some chemicals or the snake bites you, then some poisoning may happens and blood may be coagulate. This type of problems may occur. And artificial kidney. If your body does not shoot the kidney which you have submitted, which you have taken in your body by some other person, then the reaction also happens. So these are the conditions when hemolysis occurs. Now we'll discuss about the hemolysis. What is hemolysis? Hemolysis or hemolytic agents are the substances which cause destruction of RBC. So hemolysis or hemolytic agents are the substances which causes the destruction of RBC, and the hemolysis are of two types. One is chemical substances and one is substances of bacterial origin or substances found in the body. So start from the chemical substances. Number 1, alcohol, benzene, chloroform, ether, acid, alkalis, bile salt, saponin. Then the chemical poison like arsenical preparation, carbonic acid, nitrobenzene, resin. So what are the chemical substances? Repeat again. Alcohol, benzene, chloroform, ether, acids, alkalis, bile salts, saponin. Then chemical poisons like arsenical preparation, carbonic acid, then the nitrobenzene and resin. So these are the substances which can affect you and act as an hemolysin. Now substances of bacterial origin or substances found in the body. Toxic substances or toxin from bacteria like Streptococcus, Staphylococcus, Triton's bacilli. So these are the bacteria which can release some chemical substances which may affect in your body. Then venom of poisonous snakes like cobra. Then hemolysis from normal tissue. So these are the three sources which are available from the body or either from the body or either from the bacterial origin. So what are these sources? These are the source from bacteria. This is present in the body. And this is not present in the body, but it is venomous origin and it may come from the outside, which can affect by the bite of snake. So these are the points which can affect in your body. So this is our today's topic. If you have any query or any doubt related to these topics like hemolysis and the fragility of red blood cell, you can ask in the comment section. And do like, share, subscribe to my channel for supporting me and my channel. And you are always free to ask any question. Thank you so much for watching the entire video.